Section 4, we're going to talk about lymphocyte development and also antigen presenting cells. So just as a reminder, we had two types of lymphocytes, our B lymphocytes, which are a component of our humoral immunity that is going to produce antibodies that are going to bind extracellular targets. Our T lymphocytes were the component of cellular immunity where we're looking for intracellular targets. We haven't really talked about antigen presenting cells yet. We will later in this section. Just for now, just recognize that our T cells, our T lymphocytes, they can't recognize their antigen that they're supposed to without the help of an antigen presenting cell. So lymphocyte development is pretty much the same if we're talking about a T or a B lymphocyte with only one minor difference. So both T and B lymphocytes are going to originate in the red bone marrow, just like all of our formed elements, including our erythrocytes and our megakaryocytes, and they're going to stem from a hematopoietic stem cell. The next step is to undergo a maturation process, also referred to as an education process that does take a couple of days. The location of where this process occurs is going to be different depending if it's a B or a T lymphocyte. So if it's a B lymphocyte, it's going to stay in the bone marrow and undergo the maturation process. If it's a T lymphocyte, it's going to leave the bone marrow and travel to the thymus to undergo maturation. So when we talk about the maturation or education process, what we're really talking about is the lymphocyte gaining immunocompetence, meaning that it's able to recognize only one specific antigen and is only going to have one specific receptor for that type of antigen on its surface. The other component of the maturation or education process is self-tolerance. We want to make sure that it doesn't attack our own body cells, so we want it to be unresponsive to self-antigens. After we undergo the maturation process, these lymphocytes still have not been exposed to an antigen yet, so they're still referred to as being naive. So they're going to leave wherever they were undergoing the maturation process and travel to secondary lymphoid organs, which are organs like our lymph nodes or our spleen. And the whole point of doing this is that hopefully they can encounter the antigen that they have their specificity for, which is the next step. So once the lymphocytes have their first encounter, it's going to trigger the lymphocyte to develop further and differentiate into an active cell. Once it starts to proliferate, it's going to undergo what we refer to as colonial selection, where we produce huge amounts of these same cells that are capable of recognizing this specific antigen. Most of the cells that are produced from the proliferation are what are referred to as effector cells, which are capable of going in and actually fighting the infection. But remember, our adaptive immunity does have memory, so some of these cells do not become effector cells. They actually remain as memory cells, so that if we're exposed to the same antigen again, we can mount a much faster immune response. So this is just a good overview of everything we kind of talked about, the different steps in the pathway for lymphocyte development, beginning with origin in the bone marrow, maturation process, which is going to be, location is going to be unique depending on which lymphocyte it is, secondary lymphoid organ seeding, where hopefully they're going to have an encounter with the antigen that they are specific for. And if they do, it's going to cause their proliferation into effector cells and some memory cells. So a little bit more about that maturation or education process that we were talking about. There's two different things that they must undergo in order to complete the maturation and then move on to the next step of actually going to a secondary lymphoid organ. The first step is going to be positive selection process. Basically, we want to make sure that it is capable of recognizing our self-major histocompatibility complex, or MHC proteins. If they can't recognize the MHC complex as being our own, then we actually need to get rid of them, and they'll undergo apoptosis. So here's an antigen presenting cell here in purple. The major histocompatibility complex, or MHC, remember that's the protein that's actually holding the antigen itself and presenting it. Here's our T cell that's developing over here. Here's its T cell receptor. It's not fitting like a nice puzzle piece, so it's not recognizing the MHC, the self-MHC complex, which means we want to make sure that this developing T cell undergoes apoptosis. If it can't recognize our self-MHC complex, then we don't need it. Down here, we're showing that the T cell receptor fits nicely with the self-MHC complex, meaning that it recognizes it, and now it can move on to the next process. The second step is negative selection. So positive selection had to do with self-MHC complex. Negative selection is going to have to do with the self-antigens. So we want to make sure that it doesn't recognize our self-antigen, because if it recognizes our self-antigen, it's going to mark it for destruction. So here we can see that the T cell receptor fits nicely with the MHC and with our self-antigen, meaning it recognizes the self-antigen. So we don't want it to do that. So this cell is going to undergo apoptosis. If it doesn't recognize our self-antigen, meaning we don't want to go in and make a bunch of effector cells against it, then it's going to pass. So failure to recognize, which is negative selection, means that it's actually going to go on and go through the clonal selection process. So this process of maturation is actually very selective, because if not, we'll end up with a huge amount of immune disorders. That only about 2% of our T cells actually survive to become immunocompetent, self-tolerant T cells. The last thing that we need to talk about in this section are our APCs, or antigen presenting cells. So these aren't really different cells. You've heard of these before. We have our dendritic cells, if you remember from AMP1 that we talked about. So these are phagocytic cells that when pathogens enter tissues, they can go in and phagocytize it. Most phagocytes, when they phagocytize a pathogen, they basically degrade it up into little pieces, and then what they do is they put them back out to their surface, and that's why they're considered antigen presenting cells. Macrophages will do the same thing. So basically, they phagocytize whatever the foreign debris or microbe is, they chop it up, and then they display it on their surface so they can say, hey, there's something here that's not supposed to be here, and this is what it looks like. So they put all these different pieces up on the thing. B lymphocytes, we'll talk a little bit more about next, but they do not activate naive T cells, and they actually present antigens specifically to helper T cells, but it's actually to help their own activation. So dendritic cells and macrophages are our main ones that are going to present it to T cells for recognition. This is a good overview table to kind of give you the difference between the B lymphocytes and the T lymphocytes. Again, talking about the type of immune response that we've already talked about. Antibodies are only the function of our humoral response, and B lymphocytes, extracellular versus intracellular. Site of origin, which is the same for both of these, including all formed elements. Site of maturation is different. So thymus for T lymphocytes and the bone marrow for our B lymphocytes. When we're talking about the effector cells for our B lymphocytes, those are going to be the plasma cells that are going to produce the antibodies. And of course, we form some memory cells with our B lymphocytes. Our effector cells for our T lymphocytes, we have 3 different kinds. We have cytotoxic T cells, helper T cells, and regulatory T cells. We'll talk about each of these functions later on. And of course, we also form memory cells with our T lymphocytes.